Hello students, I hope you're doing fine. Now, today's lecture is going to be a very important lecture in the area of molecular biology. We'll start with the very basics of that topic, and this topic is about protein synthesis, right? So, as you all know, protein synthesis will require at least few basic types of RNA like mRNA, tRNA, rRNA, without which protein synthesis in a cell cannot happen. We are definitely going to learn about and start from those basics. But first, let me teach you about a few extremely basic points about protein synthesis. So proteins are nothing but they actually consist of 20 standard amino acid, uh, uh, and in biochemistry, you know how the naming conventions for different amino acids work. Now, I hope everybody knows that there is a basic concept in the genetic code that whatever 20 standard amino acids exist, right, they are always coded by a specific codon, right? So a codon is a set of three nucleotides which actually corresponds to one specific amino acid. We will learn that genetic code can also be degenerate in nature, and sometimes something called as synonymous codons may exist. That is something different. We will learn about that. But standard amino acids are 20 in number, and each of them may be coded by a specific codon. A codon is nothing but a set of three nucleotides that you have, right? Another important basic fact that protein synthesis always takes place from N terminal to C terminus or N terminus to C terminus. And the reading of, let's say, the messenger RNA always happens from 5 prime to 3 prime, right? So if the structure was to represent the ribosomes over a messenger RNA or an mRNA, such that there is a large ribosomal subunit on top and a small ribosomal subunit below, then the ribosomes read the mRNA in a 5 prime 3 prime direction. And when a protein will be formed, it will always be formed from N terminal to C terminus, right? Few very basic things to introduce. Now, three different types of RNA molecules are surely going to be involved in protein synthesis, without which protein synthesis cannot happen, right? And what are those mRNA? What are those specific RNA? The mRNA, rRNA, and tRNA. We are going to learn about these RNA in a sequential manner. Then only we'll be proceeding towards the actual detailed concept and steps of translation and so on, right? So let's proceed with the concept of mRNA first, right? mRNA is nothing but it carries the genetic information copied from DNA in the form of codon, right? Everybody knows that mRNA will be formed from the DNA. In DNA, you have chains, antisense strands, and accordingly, transcription happens, mRNA is formed. So mRNA will always be carrying the ribonucleotides on itself, and set of three ribonucleotide is considered to be a codon. Each codon will specify a particular amino acid, right? That will be the next point, as I said, that every codon specifies a particular amino acid, right? Now, let's have a quick comparison between eukaryotic mRNA versus prokaryotic mRNA. And in the topic of transcription, you must have heard about terms like monocistronic versus polycistronic, right? So eukaryotic mRNA are always monocistronic in nature, while prokaryotic mRNA are always polycistronic in nature. So we will understand these meanings with the help of figures and diagrams, very colorful labeled diagrams, no need to make any notes. I think the lecture in itself will be self-sufficient for you guys, right? So prokaryotic mRNA being polycistronic, you must have heard about the lac messenger RNA, right? Lac operon. So the lac mRNA containing the lac ZYA, right? So three distinct gene components are there. So lac ZYA are three cistrons, right? That's why polycistronic. And for most of the eukaryotic mRNA are monocistronic in nature. That is, they are coming from a single coding region from the DNA when they were being transcribed, right? Another important point is that all mRNA, be it prokaryotic or eukaryotic, they will always contain two types of region. And what are those two types of region? One is going to be called as the coding region, which always starts with an initiation codon, and most commonly it's AUG. And the coding region will always end with a termination codon. So coding region on any mRNA will start with the initiation codon and it's going to end with the termination codon. That's going to be called as the coding region. The other additional part on any mRNA would be UTRs, called as untranslated regions, which will never undergo translation. They will not be forming protein component. One portion being called as 5' UTR, the other being called as the 3' UTR. 5' UTR will be present towards the 5' region. They are also called as leader sequences, while the another section is present towards the 3' end of the mRNA, called as the 3' UTR or trailer sequences, right? Now, let's start with understanding the structure of eukaryotic mRNA, right? So, first point that I told you earlier also, eukaryotic mRNA are going to be monocistronic, means that they will have a single cistron. They won't be having lag ZYA or multiple cistrons like the prokaryotic mRNA. So average length of eukaryotic mRNA generally ranges somewhere between 1500 to 2000 nucleotides, or ribonucleotides more precisely. Another important thing about eukaryotic mRNA that they will always contain a 5' methyl cap towards their 5' end. A eukaryotic mRNA is always going to contain a 5' methyl cap, which will always be recognized by the small ribosomal subunit, right? When the translation is going to happen. I will show you all those steps in detail at a later point. So towards the 5' end, we have a methyl cap, as you can see. So coding region has been shown in yellow color. Starts with an initiation codon, ends with a termination codon, like initiation codon being represented by AUG in this figure and stop codon being represented by example of UA. So this yellowish region actually marks the boundary of something called as the coding region. That is actually going to form or generate a protein. While if you see towards the left hand side and right hand side, there are two extremes. Towards the left hand side, you can see 5' UTR or the untranslated region, which won't be translated, and it actually also has an initial part called as 5' methyl cap. Whereas toward the 3' UTR for a eukaryotic mRNA, you will have a poly A tail or a polyadenylation tail, and in between, stuck in between coding region and poly A tail is a 3' UTR or untranslated region, which we call as trailer sequences. This is a brief information that you should be having ideally about a eukaryotic mRNA before you actually proceed with understanding the mechanisms of translation or protein synthesis. Hope that makes sense. Now, the next thing is going to be about the structure of prokaryotic mRNA, right? So, as I mentioned earlier, also they are generally what? Polycistronic, means they will have multiple cistron like LAC-ZY, a typical example from the LAC mRNA, and I'll give you the figure-based explanation for that. Please remember, a prokaryotic mRNA does not have 5' methyl cap and a 3' poly A tail. That's not there, and that's not even required also, right? It has multiple ribosome binding site called as Shine-Delgado sequences, which we will be calling by the name short or acronym of ST sequences, right? So prokaryotic mRNA contains multiple ribosome binding sites, which are known as Shine-Delgado sequences. There are very high chances that the examinations might ask you about the the features or properties of these Shine-Delgado sequences. Please remember, the Shine-Delgado sequences are short polypurine sequences. So you know that purine is either A or G, adenine or guanine, and they are rich in those kind of sequences, right? So A, G, and U can also apply in this particular case because we are representing so this is just a single nucleotide. Otherwise, it's full of purines, A and G, or A or G, right? So it's a short polypurine sequence, right? When I will show you the structure of a prokaryotic mRNA, I will show you the location of Shine-Delgado sequences towards upstream of initiation codon. Please remember, upstream, if this is a particular mRNA and I was to show, let's say, or maybe this is my particular hand reference point. So if anything were to exist towards the left hand side of this thing, then it will be called as upstream, right? So anything which is towards left hand side from the reference point, it's called as the upstream. So when I show you the structure, I will be making their location 10 base pair upstream of the initiation codon. The SD sequences, please remember, they are complementary because the SD sequences, as I mentioned, they act as ribosome binding sites. So they will have complementarity towards the 3' end of 16S rRNA, which is a component of small subunit in case of the prokaryotic system, right? So prokaryotic ribosome, they have two subunits, large 50S and small 30S. Small 30S is the actual component which binds to the Shine-Delgado sequences, and that too because there is a complementarity between the Shine-Delgado and the 16S rRNA component of the 30S subunit. We will have quite detailed figures, lot of detailed figures and understanding of all those kind of things, right, at a later point. Now, prokaryotic mRNA also contains intercistronic region. Please remember, intercistronic region will only be found where multiple cistrons, right? Inter means between multiple cistrons. So between cistron can only be there when multiple cistrons are there. And the size of intercistronic region can vary between minimum of 2 to maximum of 30 nucleotide. Now, coming on the screen is a clear-cut picture showing a polycistronic prokaryotic mRNA, a typical example of that. If you look at the figure, there are multiple yellow regions. They are representing multiple coding regions or cistrons, and you can say they can form individual protein, like protein A from cistron A, protein B from cistron B. So they are cistron, and two cistrons will be separated by a blue region called an intercistronic region. I mean, shown with a blue arrow. And then once again, that basic structure, the left hand side area which are not translated called as 5' UTR, right hand untranslated region called as 3' UTR. Within the 5' UTR, we will be finding Shine-Delgado sequences at this particular point, and they will be very crucial for binding of the the ribosomes or the small ribosomal subunit at a later point. I hope that makes sense to everyone. Now, coming on to the next component that we have, which is crucial for translation, and translation cannot happen without this RNA, called as tRNA. tRNA, also known by the name of adapter RNA, as you must have heard. Now, the role of tRNA is basically to read the codons on the mRNA, right? So, tRNA is actually going to carry the amino acid respective to the codon of the mRNA, right? There are several examples where scientists have found that tRNA can also be involved in non-protein synthesis procedures, right? For example, it has been found that tRNA can sometimes act as a primer during reverse transcription in retrovirus life cycle, right? Can be an important question for exams or interviews. Similarly, tRNA, if we talk about the structure, tRNA is going to be a single chain ranging from minimum of 73 nucleotide to maximum of 93 nucleotide present in cytosol and the organelles of the living cell, right? Now, most commonly, there have been questions around the secondary structure of tRNA. The secondary structure of tRNA is resembling the leaf of a plant called as clover, and it appears to be similar to the leaf of that plant, and therefore it's called as clover leaf model or clover leaf structure proposed by Robert W. Hawley. And if you look at this structure carefully appearing on the screen for you, this represents what we call as the clover leaf model or the 2D structure of tRNA. So you can clearly see that we have a 5' end and the 3' end in this figure, which is containing 3 nucleotides called as ACC, right? There are several components of a tRNA. The first one on left is a loop called as D loop. Then there is a section called as D stem, right? Why we are calling it or using the word D stem? Because there is a unusual ribonucleotide called as 5,6 dihydrouridine, which is part of this particular tRNA structure, right? And then there is an unusual UG pair. You must have heard about GC pair, but this contains an unusual pairing called as the UG pair. Then when we move down, we have the anticodon loop. Then we have the stem which supports the anticodon loop called as anticodon stem. And then we have another structure called as the variable arm. Moving towards the right, we have another loop called as the T5C loop. T is representing ribothiamidine and that psi symbol is pseudouridine because these are the unusual bases present within that loop. And then we have the acceptor arm towards the 3' end, on top of which you are seeing 3 conserved nucleotides. That is, they are not going to change in most of the cases, called as ACC, right? We are going to learn about the features of the individual structure that we have just seen. So let's first talk about the D stem and loop. They are variable in length. They have unusual base called as D, capital D, which is 5,6 dihydrouridine inside them. The D stem is also containing a U and a G pair, which is unusual. Most commonly, you have GC pairing, but in this case, we have an unusual UG pair, which is present within the D stem, and these can be very important question for your examination. Coming to the next part, we have anticodon stem and loop thing. Let's start about the anticodon stem. And I have mentioned a point, it has 5 paired nucleotide. You can see there are 5 yellow dotted structures connected to each other in this anticodon stem. So 1 pair, 2 pair, 3 pair, 4 pair, and 5 pair, right? So 5 paired nucleotides in anticodon stem. Another important question for your examination. The anticodon loop is having 7 unpaired nucleotide because it's containing single nucleotide, right? And out of which there can be 2 unusual ribonucleotide called as pseudouridine and inosine, which can exist within the anticodon loop. Coming on to the next component, which is the variable arm. As the name indicates, its length can vary from minimum of 4 to maximum of 24 nucleotides. Based on its length, based on the length of the variable arm for a tRNA, tRNA can be of 2 types. Type 1, where the length of variable arm is around 4 to 5 nucleotides. Type 2 tRNA, where it ranges from minimum 10 to maximum 24, right? Now, coming to the loop towards the right hand side, which is the T5C loop, as you can see here, right? So that if I were to show you this particular loop, so this is the T5C loop that we are talking about, right? So this T5C loop, if you look at the thing carefully, it has 2 unusual bases called as ribothiamidine and pseudouridine. I hope that makes sense to everybody, right? And moving to the next component that we have is the acceptor arm. We are looking at this particular point, right? This particular area, acceptor arm. So acceptor arm has 7 base pairs, right? So these base pairs are existing here in the form of these yellow bonded structure. So 7 paired nucleotide plus 4 single nucleotide. So one here shown in yellow, which is a single. It can be any ribonucleotide, it is not fixed, but top 3 are fixed, ACC. So there are 4 single nucleotide, out of which ACC is conserved. That 4th nucleotide, single yellow dot, it can be anything, right? It's not that conserved. We talked about the 2D structure of, I would say, the tRNA, which is a cloverleaf model. And now, if I were to talk about the 3-dimensional structure of tRNA, it is something called as an L-shaped structure. Once again, important question for the examination. Please remember, each type of amino acid has its own type of tRNA, which binds to it and it carries it to the growing end of polypeptide chain if the next codon and mRNA called for it. Meaning, let's say if the mRNA contains a codon called as AUG, right? So then the tRNA containing methionine on its 3' end is going to come into picture and it will deliver methionine, right? So every amino acid has its own type of tRNA, right? So prokaryotes approximately contain 30 to 40 different types of tRNA, while eukaryotes contain approximately 50 different types of tRNA. Now, you must be thinking that I told you in the beginning of this lecture that there are 20 standard amino acids, then there should have been maximum 20 different tRNA. But how come this diversity almost double the number of standard amino acid, right? So this is basically because of something called as iso-accepting tRNA. Now you must be wondering, what isoaccepting tRNA stand for. So isoaccepting tRNA are two or more different tRNA which are specific for the same amino acid. So two or more different tRNA which are specific for same amino acid, right? So for example, if there are two tRNA specific for glycine, one of them would be labeled as tRNA glycine 1, another one would be labeled as tRNA glycine 2. Now comes the most important segment that is crucial for your examination, called as the wobble hypothesis proposed by F. Crick. Let's have a look at that. Now, the wobble hypothesis proposed by F. Crick actually explains the ability of a tRNA to recognize more than one codon by unusual, non-standard base pairing between the first base of anticodon and third base of codon, which is called as wobbling. Now, please remember, this is going to be a crucial question in the examination, and I will explain you step by step what I mean by first base of anticodon and what I mean by third base of codon, right? So ability of tRNA to recognize more than one codon. How is that possible? It is because of non-standard base pairing. Like, so what is standard base pairing? G triple bond C, A double bond T. This is called a standard base pairing. But pairings other than this kind of thing, they are called as non-standard pairs. And this is going to result in something called as wobbling. Let's try to understand how non-standard base pairing can exist between first position of anticodon and third of codon. Please remember this for examination also, right? So if I were to show you a typical example of a tRNA, let's just focus on the anticodon loop. From left side to right side, 5 prime to 3 prime on tRNA, 3 prime end is having ACC, right? So always remember, in the anticodon, towards the bottom, these three anticodon positions are written 1, 2, and 3, always based on where you are reading 5 prime to 3 prime, right? Always remember, position 1 will be closest to 5 prime end, position 2 further from 5 prime end, and position 3 farthest from 5 prime end, or let's say position 3 will be closest to the 3 prime end. So you always decide 1, 2, 3 within the anticodon loop based on how you are reading 5 prime to 3 prime actually, right? I hope that makes sense. So if I were to represent let's say a mRNA which will be complementary to this tRNA, because I've already made tRNA from left to right, 5 prime to 3 prime, I have no other option. uh, I'll have to make the mRNA from left to right, 3 prime to 5 prime, because that is called as the complementary complementarity rule, right? So one of these strands of DNA, for example, 5 prime to 3 prime, then other one from left to right will read 3 prime to 5 prime. So based on similar concept of complementarity. But I always told you that you always read 1, 2, 3, 1 to 3 positions from 5 prime to 3 prime. So on mRNA, because the 5 prime is on right hand side, you'll have to read 1, 2, and 3. So nucleotide on mRNA are 1, 2, 3, and from here to here, I mean in this direction, and 1, 2, 3 left to right on tRNA. So these things are called as wobbling position. So wobbling position is the first position of anticodon and third of the codon. They can be involved in non-standard base pairing. And in examination, question will come from this particular part. What is that particular thing? So the first position of anticodon, which is a wobbling position, and third position of the codon, they can form unusual base pairing, like meaning they can form pairing other than AT or GC. So first position anticodon can have A, then it can form non-standard pairing with U on the third position. If G is there on the first of anticodon, it can form a normal pairing with C, but it can also form pairing with U. Similarly, C here can form with G, which is a standard pairing, but U, if exists on first position, it can either bind to A or G. Then there is one more nucleotide called as inosine. It can form non-standard pairing with either A or C or U, and due to which the diversity increases, right? Means you can have two different tRNA with different anticodon and just change in position on the first nucleotide, but they might still be carrying the same amino acid on their 3 prime end. This is called as the rule of wobbling, right? I hope that thing makes sense to everybody, right? So because of these wobbling and non-standard pairing, uh, there there can be multiple tRNA which can be specific for a particular mRNA, because the pairing between first and three can allow a lot of variability, right? I hope that makes sense for everybody, right? So moving ahead, the final component that is being required is called as the rRNA or the ribosomal RNA. So ribosomal RNA, so please remember ribosomal RNA forms ribosomal protein, and rRNA plus R protein are going to result in ribosomes. They are the most abundant RNA in cell, please remember. And if you look at the ribosomes are nothing but they are the ribonucleoprotein particle which are made up of rRNA plus the R protein, and R protein are derived from translation of rRNA. Each ribosome contains two subunits, large and small. Ribosomes always read the mRNA from 5' to 3' direction. Let's have a look at prokaryotic ribosome. Prokaryotic ribosome have two subunit every, that's a universal rule. So large subunit is called as 50S, small subunit is called as 30S. Now in your examination, a breakdown of these components is going to come. So 50S subunit has 23S rRNA, 5S rRNA plus 31. That's the number or count, 31 ribosomal protein. While 30S is going to have 16S rRNA plus 21 ribosomal protein. Eukaryotic ribosomes, if we look at the large and the small subunit, so the large subunit is called as 60S, the small subunit is called as 40S. Large subunit will have 28S rRNA, 5S rRNA, 5.8S rRNA, and 50. Once again, that's the count, 50 ribosomal protein. While the 40S subunit is going to have 18S rRNA plus 33 ribosomal protein. I hope that makes sense to everybody. This is about basically the composition of, I would say, the three different types of RNA molecules which are crucial for the process of translation, right? And in our next lecture, we are going to start with the role of rRNA in translation, and then we will essentially start with the process of translation and mechanism. So that will be all for today. Hoping to see you in the next lecture, and I hope you like the style of delivering this lecture. The content is clear cut, easy to understand interpretation and diagram, and very soon we'll be coming with the next part of protein synthesis. See you in the next lecture. Thank you for your time and patience. Best of luck.